Today's topic for discussion is life cycle of Marsilea. Marsilea, it is a pteridophytic plant, and Marsilea, it is the member of order Marsiliales. This order includes a single family Marsiliaceae, and this family that include living genera like Marsilea, Pillularia, Regnellidium, etc. And the Marsilea, it is a heterosporous plant because that produces two types of spores. The sporangia of these are produced within special structures which are known as Sporocarp. The spore producing structure of Marsilea is known as Sporocarp. Each Sporocarp that possesses many sori which bear microsporangia and megasporangia. The sori are gradate. Sori, it is a group of spore. That is, the oldest sori are found at their terminal end of the receptacle and the youngest at the base. Let us see the details of only Marsilea. There are about 65 species of Marsilea which are distributed throughout the world. Marsilea occurs throughout the world. They are more commonly found in tropical regions such as Africa and Australia. In India, 10 species of Marsilea was discovered by Gupta in 1957. About 6 species of Marsilea are known to occur in United States, and they are hydrophytic or amphibious plants. That means they grow near water or they can grow in water as well as on land. Such plants are described as amphibious plants. They grow rooted in the mud of marshy places. And Marsilea vegetativa, it is one of the species, and some other species grow in shallow ponds. A single plant, Marsilea vegetativa, may grow in all directions until it covers an area 25 meters or more than that. The Indian species recorded by Gupta and Bhardwaj in 1957, the species of Marsilea which occur in India, they are Marsilea quadrifolia, Marsilea minuta occurs in India, Marsilea aegyptiaca, and Marsilea brachypus, Marsilea gracilenta, Marsilea punensis, Marsilea rajasthanensis, Marsilea brachycarpa, Marsilea condensata, and Marsilea coromandelica. These are the species of Marsilea which occur in India, that is recorded by Gupta in 1957. Let us see the external morphology of Marsilea. Let us see the stem first. The species of Marsilea that possesses rhizome which creep on the soil surface. The rhizome is cylinder, branched, and that is having nodes and internodes. Rhizome possesses nodes and internodes that grow horizontally on the soil surface. The leaves are borne alternately along the upper side of the rhizome, usually at the nodal region. One or more adventitious roots that grow out from each node from the lower surface of the rhizome. The rhizome is dichotomously branched and it is capable of indefinite growth in all directions and cover an area more than 25 meters in diameter. Let us see the details of leaves. The leaves are borne alternately along the upper side of the rhizome at nodes. The leaves possesses sarcinate venation typical of most follicles. The young parts of the leaves of Marsilea minuta and others are covered with numerous multicellular hairs. The leaves are leaves of submerged plants that possesses long flexible petioles and leaf lamina that float on the water surface. When the plants grow on mud or marshy places, the leaves have got shorter and petiole erect and stouter, which spread the leaves in the air. The leaves are compound. The leaves of Marsilea are compound. The lamina of each leaf is divided into 4 leaflets. The leaflets are also known as pinnae. Division of leaf blade into 4 leaflets result from two dichotomies arising in close succession of each other. Okay. Let us see the sporocarp. Sporocarps are borne on short peduncle near the base of the petiole. Sporocarp, it is the spore producing structure. In majority of the cases, the peduncle or the stalk of the sporocarp is unbranched and bear a single sporocarp at its apex. But in some species of Marsilea, like Marsilea quadrifolia, the stalk of the sporocarp is dichotomously branched, which bear 2 or 5 sporocarp. In this way, in Marsilea polycarpa, single dichotomously branched peduncle bears about 6 to 26 sporocarps. Let us see the external morphology of roots. One or more adventitious roots are borne at each node on the underside of the rhizome. The adventitious roots are supporting roots. The adventitious roots may arise even from internode in case of Marsilea aegyptiaca. In case of Marsilea minuta, the roots may develop laterally. The roots of Marsilea minuta secrete carbonic acid and because of this type of secretion, the roots could penetrate the shells of the pond snail also. This is the external morphology of Selaginella plant. Let us see the transverse section of the stem. Let us see first the transverse section of rhizome of the Marsilea. This is the photograph shown for the rhizome of Marsilea. Rhizome of Marsilea that possesses amphiploic syphnostele. It is a characteristic feature. And the outermost layer is single layered, which is known as epidermis, without any stomata. Stomata are absent in epidermis. The cortex is placed below the epidermis and the cortex is differentiated as outer cortex and inner cortex. The outer cortex that consists of compactly arranged parenchymatous cells. This region may be several cells in thickness. Just below this, the cortical region contains large lacunae or air spaces or parenchyma. This is also a characteristic feature. The lacunae are separated or the parenchyma are separated from each other by a layered parenchymatous septa, and this region may be considered as middle cortex. Internal to the inner cortex, there is a zone of sclerotic tissue. The cells consisting this region are thick-walled. They are sclerenchymatous in nature. Beneath this region, the cortex again consists of compact parenchymatous tissue acting as storage tissue containing starch in them. Some of tannin cells may be found in this innermost region. Let us see the stele of the Marsilea. The stele is amphiploic syphnostele. In case of rhizobium of Marsilea, all species possesses the syphnostele externally and internally limited by single-layered endodermis. Endodermis is single-layered. Just beneath the outer endodermis, there is a single-layered pericycle. In the center, there is a pith, and in submerged species, the rhizome possesses usually parenchymatous pith, whereas plants growing in mud have a more or less sclerenchymatous pith. Okay, next. This is the photograph of petiole. Let us see all the details of the internal structure of petiole. The internal structure of petiole as seen in transverse section in the photograph. The outermost layer is single layered epidermis. Just beneath the epidermis there is a hypodermal layer. Then there is outer cortex constituting a large air spaces which is known as parenchyma, which is separated by septa. The inner cortex is composed of parenchymatous cells. In this region some of the tannin cells may be present here and there. In the central region of the petiole there is a stele which is more or less triangular in outline. The stele is surrounded by single layered endodermis. It possesses V-shaped mass of xylem with the exarched protoxylem. The two arms of V, capital V, are quite separate and somewhat curved as shown in the photograph, and the V-shaped mass of xylem is surrounded by phloem and single layered pericycle successively. The ends of each arm of the V-shaped xylem are protoxylem groups, whereas the middle region of the arm of V consists of a large tracheids representing the metaxylem groups. The opening of V is always toward the axis, that is adaxial side. Next, this is the photograph of vertical section of leaflet of the Marsilea. Let us see all the details of internal structure of leaflet. As shown in the transverse section of the leaflet, it is bounded by upper and lower epidermal layer. In case of floating leaves, the stomata are confined to the upper surface, but in case of plants growing on the mud or muddy soil, the leaves are aerial and the stomata are found on both surfaces, upper and lower surface. Stomata are present in both the epidermal layers of the land plant. And the mesophyll is present below the epidermis. The mesophyll is differentiated into palisade and spongy tissue. The vascular bundles are concentric. That is, concentric vascular bundle means the xylem elements are surrounded by phloem. On the outside of the vascular bundle, there is a single layered epidermis. Towards lower side, there are big air spaces separated by septa. These are the details of vertical section of leaflet of Marsilea. Let us see all the details of the root. This photograph is shown. This is the transverse section of root of Marsilea minota. In the transverse section of root, the epidermis, cortex and stele may be seen. The epidermis is single layered and composed of thin walled parenchymatous cells. The cortex of the root is differentiated into two regions. The outer region that consists of large air spaces which are separated by septa from each other. In the inner zone of the cortex that consists of few layers of sclerenchymatous cells. In the cortex there is a single layer of epidermis. Just below the epidermis, a single layered pericycle is present. In the center of the stele there is a typical diarch and exarch. Diarch means two vascular bundles of two elements of protoxylem present, that is diarch and exarch structure with diametric plate like strand of xylem is present. The phloem bands are found on either side of the xylem plate. Next, let us see the reproduction in Marsilea. The reproduction in Marsilea takes place by vegetative and asexual method. Vegetative reproduction, let us see the vegetative reproduction. The vegetative reproduction in Marsilea takes place by forming tuber-like structures. In the dry conditions, certain branches develops from the rhizome which swell because of the storage of the food material, and these structures are known as tubers. These tubers release after maturity and independently produce a new Marsilea plant in favorable environment. This is about the vegetative reproduction in Marsilea. Let us see the sexual reproduction in Marsilea. The plant is sporophyte, that means spore producing plant. It bears the spatial structure which is known as spatial structure for production of spores, which is known as Sporocar, And the Sporocar produces two types of spores, that is microspores in microsporangia and megaspores in megasporangia. Therefore, it produces two types of spores. Therefore, it is a heterosporous plant. The sporocarps are borne on a short peduncle above the base of the petiole. In most cases, the peduncle is unbranched and that bears a single sporocarp. The sporocarp may be oval or bean-shaped. In the earliest stage of its development, it is soft and green, but later on it becomes sufficiently hard and brown in color. In majority of the species of Marsilea, near the point of attachment of the stalk, these are usually one or sometimes more protuberances in the median plane takes place, and that develops into a sporocarp. And this is the internal structure of sporocarp. To study the internal structure of sporocarp, the sections cut into longitudinal and transverse plane at right angle to the flat surface is required. The internal structure of sporocarp is transverse, longitudinal and dorsiventral section as shown in the photograph. Let us see all types of sections. Let us see first the transverse section. The following tissues are seen in the transverse section. Outside the sporocarp, there is a very thick wall that consists of three layers. The outermost layer is known as epidermis, which consists of broad and columnar cells. Epidermis is interrupted by a number of stomata. Below the epidermis, there are the hypodermal layers. The outer hypodermal layer that consists of elongated thick-walled cells, while the lower hypodermal layer consists of elongated and palisade-like cells. In the inner region of the sporocarp, only two chambers of sora are seen, surrounded by separate indusia. The dorsal bundles are also seen. Each sorus possesses a receptacle which bears either micro and megasporangia. This is the vertical section or longitudinal section of the sporocarp. Let us see the details. The structure of the wall of sporocarp and the gelatinous ring is the same as in transverse section. In the interior of the sporocarp, there are two alternating rows of sori, each surrounded by two layered indusium. A placental branch goes to each sorus from the lateral bundles as shown in the photograph. The receptacle bears a single megasporangium at the tip and microsporangia on the lateral side. Each megasporangium contains a large single megaspore, while the microsporangium contains many small rounded microspores as shown in the photograph. This is a sporangium, developing sporangium. These are the details of sporangia. This is gametophytic generation. Let us see the gametophytic generation in next slide. Okay.